Hello, everyone. I would like to get started. It is both a privilege and distinct pleasure to welcome you all to the first Pediatric Neurology Grand Rounds Lecture of 2023. Dr. Francisca Hosch invited this speaker, Dr. Zollei, and so I'm speaking on her behalf because she's lost her voice. Today, we have the honor of hosting Dr. Zollei, an outstanding researcher whose work has changed the field of how we see the developing brain. Dr. Zollei is an associate professor of radiology at the MGH and assistant in neuroscience at the MGH. She holds a secondary appointment in newborn medicine at Boston Children's Hospital. She received her Master of Science and PhD in computer science from MIT, followed by postdoctoral research at the École Centrale de Paris in France. Sorry for the mispronunciation. Dr. Zollei's work focuses on various neuroimaging solutions related to human brain development. She is interested in MRI brain image analysis during the perinatal period and writes tools to analyze both postmortem and in vivo high-resolution images. She enjoys collaborating with people from various scientific backgrounds. Her research interest includes, but is not limited to, algorithm development, brainstem, cerebellum, cerebrum, consciousness, diffuse magnetic resonance imaging, diffuse weighted image alignment, human brain development, image processing, computer-assisted imaging, three-dimensional imaging, in the infant and newborns, longitudinal analysis, MRI, multimodal neuroimaging, pattern recognition, and automated pattern recognition, and postmortem brain tissue analysis. She is the recipient of multiple NIH grants on this topic, awards of academic excellence, and the Ankobansky Visiting Scholar Award. Her research funding includes multiple R awards for investigation of longitudinal atlas construction of brain development from ex vivo to in vivo prediction of the developing human cerebellum, uniting computational engineering and neuroscience fields to advance neuroimaging discoveries for the young child. She's also developed the Infant Free Surfer, an automated infant brain MRI analysis toolset to accelerate infant neuroimaging pipelines, including ease of use and cloud readiness for a worldwide user base. Dr. Zollei's work reminds us all that medicine is not just a science, it is a lifelong journey of discovery, compassion, and service. Please join me in extending a warm welcome to Dr. Zollei.
Thank you very much for this warm welcome. And I'm going to get started. First, I have a couple obligatory slides for you. No disclosures. So today, in the upcoming talk, I'm going to tell you a little bit about the various projects that my lab is working on. And I'm going to get started with the in vivo MR analysis toolset. So the overall motivation for this section can be summarized as follows. I don't need to describe, but MRI is a powerful and flexible tool for characterizing neurodevelopment. It can visualize and quantify brain structure and function, and it's safe and non-invasive and can provide quantitative measures of brain growth and maturation at high spatial resolution. However, there are unique challenges of conducting MRI in children, particularly the very young ones, and other challenges when analyzing their data. Just to show you some of these complexities, I'm going to use brain scans that were required on a single subject at birth, 1 year and 2 years of age. In the upper row, you have T1 weighted scans of the same subject. The bottom row is T2 weighted scans, and the bottom row is the segmentation or labels that were required using these scans. So I would like to point out, first of all, the size differences between the newborn and the 2-year stage. So there's the brain size almost doubles during this period. So our tools need to be robust enough in order to handle such changes. There's also a contrast with MRI images. So if you look at the newborn stage and then the 1 year or 2 year stages, image intensities that were dark in case of the newborn scan flip into a lighter one, and darker ones flip into a lighter one. So again, this has to be accommodated. And of course, there's the development. So secondary folding patterns are still developing. Myelination is still taking place. So again, the tools that are adapted or developed for this particular age period need to be flexible enough and robust enough to handle all of these characteristics. So the first tool that I'm going to introduce to you is Infant Resurfer. It's an image processing pipeline that has been around for a while. So this is not our newest tool set, but this is going to demonstrate all the different aspects and the key computational blocks that we have to focus on in order to be able to quantitatively analyze these images. So the goal when developing this tool set was the following. We wanted to have a tool, an algorithm that will be able to provide us with a full brain segmentation, including subcortical and cortical segmentation, and also present us with a surface representation of the brain. And I'm going to tell you more about the importance of that. We wanted to use the FreeSurfer format. This, if you happen to be in the neuroimaging domain, you know that it's a tool set that has been around for more than 2 decades for the analysis of adult brains mostly. And the age range of interest for my tool development was the first 2 years of life, given all the challenges that I presented to you before. The original version of Infant Resurfer was designed to work with T1-weighted MRI scans. And the 3 main computational blocks that I would like to highlight today are listed here. So here's the pipeline, kind of to guide you through visually what happens. So we have an input image, a T1-weighted input image. Then we do some pre-processing to it. Then we do the first larger computational step, which is skull stripping. So we basically erase or get rid of anything around the brain that's not part of our region of interest. Then we do volumetric segmentation or label key anatomical areas in the brain. And the last largest computational block is creates surfaces. So these surfaces, we create 2 surfaces on each of the hemispheres. One is called the gray-white or the white matter surface, and then the other one is the PL surface separating the gray matter and CSF. When the surface representation is ready, then we can also label cortical areas on the surface, and that's what we call parcellation. So very quickly, I'm going to tell you quickly about our solutions to these computational blocks. So in case of our skull stripping tool, we use the supervised computational neural net. We have a large training data set. We have representatives from all of the ages with their manually segmented brain masks. And we train 3 independent 2D fully convolutional neural nets in order to get a representation of our training data set. And at prediction time, what happens is when we have a new input image, we get 3 different solutions predicted by these networks, and then they get linearly fused together, thresholded, and we have our brain mask as the output. Now we have our brain mask, so we have our region of interest. Now what we want to do is assign different anatomical labels or segmentational labels to them. The original tool or the idea that we had was to use a prior-based spatial approach in order to do that. That requires a training data set again. And for this task, we had 26 manually segmented MRI images, and we tried to have them span almost uniformly the age range of interest. The tool that or the solution that we implemented is a multi-label atlas fusion concept. And that allows us to combine training data with potentially missing labels. And I'm going to show you in the next slides that sometimes labeling the gray-white boundaries was not possible in this data. So having this particular solution was very helpful. This framework also does not make assumptions about intensity distributions, which is really helpful given all the variability that exists in our data sets. So let me show you a more conceptual visualization of how this framework works. So we have our training data set. We have 26 of these images, and we have their corresponding manual segmentations. When I have a new image that gets acquired and needs to be segmented, we do the pre-processing and the skull stripping. And at this stage, we either take a subset or the full training data set and spatially align or find correspondences between this training data set and my test image. If I find a correspondence between the training data set, I can also bring the manual segmentation into the same test image domain. And doing some heuristics and mathematics, I can come up with a segmentation solution for that particular brain. To give you an example, I'm showing you segmentation solutions that got acquired from a newborn subject, 8-month-old, 12-month-old, 16-month, and 18-month-old. So showing you the robustness of the tool throughout the whole age range. In the first column, we have the T1 weighted input images that already got skull stripped. In the middle, we have the manually segmented solution, and on the right-hand side, you see the automated solution, which is very closely matching the manual solution. The third step, as I mentioned to you, is the surface extraction. This is going to be computationally very important, and in the background, we use that a lot, especially when we are trying to characterize the cortical ribbon, functions that can be defined in these areas. We're trying to describe, for example, cortical thickness. We initialize the white matter surface placement with our volumetric white matter labels. Then the PL surface is going to be initialized and grown out of the surface. And through computational or algorithmic tools, we are going to find the boundary between the gray matter and the CSF. Throughout this process, we are also carrying out topological correction in order to make sure that our surfaces are mathematically correct and well represent the surface. So this is a typical representation of the PL surface that we acquire. So here I'm showing you a newborn, 12-month-old, and an 18-month-old subject. The color coding is very specific to free surfer, so it might be strange, but it's presenting you with some cortical measurements painted on the surface. In the middle row, we have cortical parcellation labels projected onto this surface. And in the bottom row, this is a representation that's rarely gets seen by the users, but it's used very heavily in the background. So this is the same surface that you see on the top, but projected onto the sphere of the surface of the unit sphere. And mathematically, it's a 2D domain that's going to be much easier for us to do computations in. As I mentioned, this code has been around for a while. You can download it, play around with it. Please send me an email if you have any issues utilizing it. And very recently, in order to increase accessibility and also reproducibility, we have also developed container-based methods that carry out or help you run the tools. So hopefully with that, it's even easier to run any of your experiments. Now, I introduced you the main computational blocks, but I want to give you a little bit of an insight into how my collaborators are using these tools and what interesting projects we have been working on the user side. So one key area where this tool gets used is population analysis and population studies. The example that I'm going to be using is a large-scale data collection and data analysis project that was led by multiple teams all around the United States. So the main hypothesis was the following: the human brain grows quickly during infancy and early childhood. However, factors influencing brain maturation in this period remain poorly understood. So in order to address this gap, the team harmonized data from 8 large and diverse cohorts, creating the largest pediatric neuroimaging data set to date, focusing on the age between birth and 6 years of the interval. The team mapped the developmental trajectory of intracranial volume and subcortical volumes in about 2,000 children and studied how the socio-demographic factors and adverse birth outcomes influence the brain structure and cognition. So the type of graphs that you can get when you have quantitative measures in order to help this analysis are follows. So I'm showing you an example where we have the intracranial volume mapped by age. On the right-hand side, you see all the data sets that participated in this study. Most of them come from the US, but there's one from Singapore and one from Cape Town as well. So some of the main conclusions, and this paper is under review currently, are the following. The amygdala was the first subcortical volume to mature. The thalamus exhibited protracted development. Males had larger brain volumes than females. Children born preterm or with low birth weight showed catch-up growth with age. The socioeconomic factors exerted region and time-specific effects. And regarding condition, male region-specific scored lower than females. And the brain cognition correlations revealed region-specific associations. Another very exciting use case scenario is developing numerical models. So with a collaborator at the Martino Center, Dr. Bonmassar, we worked on creating open source computational models with full body anatomical details. Dr. Bonmassar's group developed two different models, Martin, who is a 29-month-old male subject, and Athena, who is representing a 42-month-old female subject. These numerical models can be used for investigating tissue interactions with electromagnetic fields generated from various medical devices, and for MRI RF dosimetry studies of age-matched children. Athena, the female subject, was also the first toddler used for MRI safety example in a 70-head MRI. And given that we have both a male and female model, we can also study sex as a biological variable in our studies. Again, just to show you with more images of what went into creating these models. So retrospectively, two subjects were selected who have images covering the full body. These are MRI and CT sequences. My tool participated in the automated analysis of the brain images, and then it got incorporated in the full body segmentation, which was very heavily manually done. And by showing you the details, you can appreciate all the work and detail that went into it. All right, so the next tool that I want to show you is a more recent tool, and it's called a fast infant free surfer. Traditional tools, including the infant free surfer model that I just showed you, can be quite time consuming, especially when we are computing spatial correspondences that are often nonlinear transformations. They also often require resampling to a common resolution, which means that we lose a lot of information from our input images. So we wanted to have a tool that's going to address these issues, and we turned to a deep learning solution. And the tool that I'm going to introduce is specifically focused on accommodating two challenges. One is that the infant data sets that we see have large variations in head positioning. So even though we are doing our best in order to have a standard orientation or head positioning for the infants, oftentimes we see images with all sorts of different orientation in the acquisitions. And also, even though we are lucky that recently there has been more and more commonly shared large data collection projects funded, there's no standard resolution for these images. So any tool that we are developing in order to analyze these images need to be able to work with all sorts of different resolutions. So when we turn to deep learning solutions and we have limited amount of data sets, and in case of infant brain imaging, we are always working on a very small scale, small data samples. We need to do something which is called augmentation. Augmentation means that we are trying to increase the size of the training data by, for example, scaling, rotating, and translating the already existing data set. So kind of imitating a larger training data set. So convolutional neural nets, they need this external augmentation in order to be robustly functioning. There's a recent tool called VIN, or voxel size independent neural networks, which was developed by a former Martino's colleague, Dr. Reuter. And this tool managed to migrate at least some part of these transformations or augmentations to within the model. So the scaling is inside the model, however, it still needs some external augmentations. And the problem is that both of these deep learning solutions or frameworks still require resampling the label, so we are still losing information when we are carrying our computations. So our solution was to develop a model or design a model where all of these augmentations happen within the system, not outside. The tool is called VINA, or Win with Internal Augmentations. We have 4 degrees of freedom from the transformation module, and this resulted in a non-lossy label interpretation. And by implementing this model, this led to less information loss when we do our computation. Let me show you the images that we are working with and the type of solutions that we get with this newer approach. So on the top, you see one particular subject from the DHCP dataset. DHCP stands for the Developing Human Connectome Project, which is a project that was funded in the UK. Most of its subjects have both a T1 weighted and T2 weighted image, and the group also released a set of segmentation labels accompanying all of their acquisitions. So this is going to be the one that we are using as a ground truth. In the bottom, under predictions, we are showing you state-of-the-art methods that are segmenting these brains. In the middle row, we are segmenting T2 weighted images, or tools that are segmenting T2 weighted images, and then the bottom, these are T1 weighted based segmentations. So in the bottom left, we have infantry surfer, for example, and VINA can work on both modalities, both T2 weighted and T1 weighted images. So we have solutions from it in both of those rows. I just want to point out with these little arrows and zoomed in versions of the solutions that VINA manages to see, manages to preserve structures better, which is going to be very important when we are creating surfaces and trying to quantify, for example, cortical thickness measurements. So in summary, this is a new tool that we recently developed. It's appropriate for newborn brain segmentation and produces 89 labels, both subcortical and cortical labels. It supports native resolution segmentation. So depending, basically the segmentations are going to be matching your input resolution. And this new solution outperformed both traditional and deep learning models. Very quickly, I'm going to spend a little bit of time on showing you another algorithmic solution that focuses on the surfaces. So I mentioned several times that computing or having a representation for the cortical surface for us is very important, because we can do more accurate measurements and compute correspondences between different brains using it. So I mentioned also that there's a rapid development of cerebral cortex during the perinatal period. Hence, any type of static atlas is not going to be able to represent these brains during the period. So we had to develop a spatiotemporal atlas that can actually represent this time development or age axis of this process. We also have to face the fact that there's limited availability of both data sets and atlases around. So we came up with a new solution. And what we propose is another deep learning methodology. It's a deep neural network that achieves end-to-end training for group registration and also produces at the same time a temporal atlas that well represents our training data set. Again, visually, I'll show you, this is the Atlas construction framework. I'm not going to go into the detail. I want you to focus on, it has two different components, an Atlas inference component and the registration or a spatial correspondence computational component. So here A is an important feature. In our case, this is going to be a feature which represents age, but it can be sex or any other type of variable that you want to define or differentiate your atlases with. So this Atlas inference framework outputs a representation of your training data set given that particular feature. And if you have if you have a test subject that you want to align with this atlas, our sphere morph registration solution provides this close correspondence between them. And now you can use this mechanism in order to transfer information from your atlas space, for example, parcellation labels, into the subject that you are going to be analyzing. Maybe a little bit more intuitive representation of how the output looks like. So after training our models, we can generate a specific convexity atlases. So for example, this is a newborn data set that we used, the DHCP cohort, and we were able to generate atlases representing time points from 35 to 43 weeks. Note that this is a continuous representation, so even if my training data set didn't have a particular age, this tool is able to generate you a representation filling in the spots in this age range. I zoomed in on a particular area in order to focus on the changes in a certain area, which allows us to suggest that there's longitudinal consistency in our representation. This is cross-sectional data, but we believe that we do see the atlas picking up these longitudinal developmental patterns. Again, it's very hard even for people who are actually developing these atlases to characterize how good an atlas is. So oftentimes the goodness or the performance of an atlas is characterized by a follow-up application, and that application is usually a segmentation. So here we did exactly that. We used our templates in order to guide parcellation or surface spherical segmentation of a subject. So we have a cohort for which we have manually segmented labels on the surface. We use our tool and one of the other existing methods in the literature in order to align these frames with an atlas that contains all the parcellation labels, and then was able to measure accuracy and compare how well our tool is working compared to the manual segmentation. And the last tool that I wanted to introduce to you switches imaging modalities, and that can be applied or applicable to white matter imaging. So you well know that we cannot image individual axons. Their size is on a micron side, but these axons bunch together and they form bundles and they traverse the white matter. So diffusion-weighted MR images help us image these white matter bundles, and these images are very useful in studying neurodegenerative diseases, stroke, aging, and development. So diffusion-weighted images differentiate tissue types based upon the diffusion of water molecules within them. So for example, in the gray matter, diffusion is uniformly restricted. So this is an isotropic movement. And in case of white matter, diffusion is preferentially restricted. So this we call anisotropic. So instead, if we have an image, and here I'm showing you the simplest model that can be applied to reconstruct a diffusion model. Instead of having an image intensity value at a coordinate location, we have tensors representing this particular diffusion movement. Now, just like in case of the structural images, we want to be labeling these fiber bundles, and there are different methods that can be used for that. Manual dissection is, one methodology, but it's very time-consuming and operator-dependent. So there are automated tools that are atlas-based, but they heavily rely on alignment, just like the previous tool that I showed you. So a colleague of mine, a little bit more than 10 years ago, Dr. Yandiki from the Martino Center, came up with a system called Tracula, which uses not only diffusion-weighted information, but also anatomical information coming from free-surface segmentation labels in order to guide the placement of global tractography solutions. So we wanted to use the same framework. So we used the overarching framework, however, had to redefine all the individual components, and in particular, the training data sets that went into this tool set. So hence the development of Traculina, which is a tool applicable particularly to newborns. And the training data set here used 14 different white matter fiber bundles segmented on 30 volumes, and the initial segmentation was done on deterministic tractography. There was a recent development in Dr. Yandiki's group where they showed that with high-quality multi-shell diffusion MRI data, if we resegment or relabel the tract solutions and use the same Traculina framework, we are able to more robustly and accurately label even low-resolution single-shell data sets as well. So we definitely wanted to have that for the infants as well, given all the motion and inaccuracies that we still see in our infant solutions. So the new training data set for Traculina included 15 subjects. This was in collaboration with Washington University, who provided us with multi-shell high-quality diffusion MRI data sets. We did our usual diffusion pre-processing before the segmentation. We also ran inference resurfer in order to get to the anatomical labels, align these images spatially, and did the manual segmentation of now an increased number of white matter bundles, 26 of them. And after we did that for the whole training data set, we were able to show via a Levonat cross-validation experiment that indeed, in case of the newborns as well, accurate and robust measurements and labeling can be acquired by using this high-resolution data set, even on single-shell data sets as well. All right, now I'm going to switch gears a little bit, and I'm going to talk about collaborative infant imaging. And this is something that Dr. Hosch, pointed out that she has interest in. And so, in order to talk about this project or this collaboration, I want to introduce you to a society. This was recently formed, and I was one of the founding members in 2018. This is the Fetal, Infant, and Toddler Neuroimaging Group. This society welcomes interdisciplinary scientists and clinicians with an interest in early neurodevelopment and encourages collaboration and innovation. We have close ties to the NIH and the A3CD study, and we put a key emphasis on the establishment and dissemination of guidelines and support best practices for methods used to study the developing brain, including various types of imaging modalities. We have just finished our second independent conference in Santa Rosa, and here I'm including the URL to the society. If you have any questions, please ask me, but this is a very active group, and we would welcome any of you to join. So as I mentioned, the society is very active academically. We write grants, and we currently have a grant that allows us to organize conferences and workshops. But we have also, in the past 2 years, have published various collaborative team-based papers that are addressing key issues that we are facing as a community in pediatric neuroimaging. The paper that I wanted to spend a little bit of more time on describes the art or science of scanning young children. As I mentioned in my introduction, MRI is a very useful modality. However, not only the analysis of their data, but acquiring MRI images can be quite challenging. And this paper describes suggestions and areas where we can improve in order to increase the success rate of successful imaging in this population. So the 4 areas where innovation or improvement can help, with increasing the success rate that was identified by this paper are the following: synergism with clinical efforts, flexible family-focused protocols, community engagement and education, and optimized hardware and software for our subjects. I'm going to give you a little bit more specific examples. So major challenges when we are scanning this population are the following. Motion is always something that we are constantly fighting. So many studies are doing their scanning under general anesthesia, sedation, or wait for a natural sleep state. In parallel, on the technology side, there has been a lot of efforts put towards motion correction, both prospective and retrospective. My colleagues at the Martino Center are heavily involved, for example, in the prospective motion correction work. The scan duration, the scan time can also be very anxiety-inducing or very limiting for some of these subjects. So the development of shorter sequences that are using undersampling or simultaneous multi-slice imaging is also very prevalent. Noise levels can also be very high, especially with the various different types of MR images that we are using. So besides developing quiet and silent imaging sequences, the use of headphones and earmuffs has also been highly recommended. And equipment sizing is also quite key. So collaborators at the Martino Center have been developing custom-built developmental head coils that are better fitting of the smaller subjects. So this example here is a picture of a coil that was developed for Dr. Rebecca Sex's group at MIT by people from Dr. Larry Wald's group at the Martino Center. As I mentioned, scan sessions can often last up to 3 hours, which is very long. So additional things that can help make these sessions more successful are the following. So there's improvements that can be made in how subjects get transported, what type of additional furniture that can be placed in the scanner room for parents and for research staff in order to accommodate subjects and babies during the scanning. Or we have traveling cribs these days that also accommodates kind of transporting sleeping babies, in and out of the scanner. We have also identified that sending out informational material, training samples, for example, of the noise level that children can expect during the scanning can help reduce anxiety that's related to the scanning. And also hiring, so to speak, baby whisperers or research staff that can be very understanding and can address all the questions, coming from the families, parents, and the accompanying staff with the children can be very, very useful. Other interactive material, for example, books. Here I'm showing you an example that's provided by Siemens in multiple languages. Helps kids, learn about, what happens in the MR scanner, or doll-sized models, Lego sets that again help kind of describe how this large magnet is not a scary machine and how to keep them a little bit, more relaxed in the scanner. One very specific example just recently got presented at our fitting conference, and this was done by a former colleague, Melanie Gans, who's currently working in Copenhagen. And they have done the following. So after doing a careful and detailed analysis of retrospective analysis of motion during infant scans, they designed a clinical imaging protocol in the hopes of reducing motion artifacts and increasing the success rate of their scanning without general anesthesia. So the protocol has 3 different steps. The first step includes preparation with a mobile app. Here, this little white bear explains the whole experience to the subjects and parents. Then there's a mock scanning training, which takes place in the hospital. Again, the children are distracted with movies, and their movement causes the movie to either shrink or disappear, so encouraging them to lay still. And during the mock scanning, we are also doing head motion tracking in order to quantify the motion that's anticipated. And during the actual MR scan, the distraction of the children with the movies still takes place. There's head motion tracking and also prospective motion correction. What they've been able to show on a cohort of 47 subjects with an average age of 7 is that this preparation or this multiple framework helped to reduce the rate of general anesthesia by 94%, which is huge. And they were also able to show that prospective motion correction and selective reacquisition is feasible in a fully clinical pediatric MRI cohort. So the last set of projects that I wanted to introduce to you focuses on ex vivo imaging of the perinatal brain. The motivation for this project was the study of SIDS. And SIDS is the sudden unexpected death of seemingly healthy infants under the age of 1, where the cause of death cannot be determined by autopsy or the death scene. This is unfortunately still the leading cause of postnatal infant mortality in industrialized nations. And according to the triple risk model, there are 3 factors that need to be present for SIDS to occur. The infant needs to be in the critical developmental window. It needs to be a vulnerable infant, and there has to be an exogenous homeostatic stressor as well. The leading a hypothesis that was leading our kind of research interests and the definition of our project is the brainstem hypothesis. And it states that there is a developmental abnormality in this region that leads to a failure in the protective mechanisms in place against life-threatening events during the sleeping period. The brainstem, as you well know, is the main anatomic site of sleep and waking regulation, and it also has the homeostatic controls. Postmortem psychological studies have shown abnormalities in its serotonergic systems, and what we are trying to replicate and study further is the same type of abnormalities, but with high-resolution 3D imaging modalities. And note, there's no anatomical biomarkers currently that are available to identify living infants at the risk of SIDS. So our goal in our project in this ex vivo domain is the following. We wanted to image and map the neuroanatomic connectivity of the ascending arousal network in postmortem fetal and infant brains. We also wanted to establish the correlation of SIDS with high-resolution imaging, and the imaging modality here is ex vivo MRI and optical coherence tomography, or OCT for short. And also, being a tool developer, we wanted to develop tools that can automatically segment, register, put into alignment all these different data sets that we are acquiring in order to be able to more accurately and robustly analyze a larger cohort of these images. This is the top-level kind of summary or representation of the different stages of our project. So we have established a tissue bank, which contains full brains that have been fixed. Then we have a bunch of different imaging steps, starting with whole brain MRI, then a blocked brainstem MRI, OCT, and histology at the very end. So we get our tissue samples in our brain bank, it gets prepped, fixed, and then we get it ready for MRI imaging. And then we start the imaging protocols. So here I'm showing you a 100-micron resolution scan that was taken of the brainstem itself. It's very high resolution. If you think about it, usually in clinic, you see 1 millimeter isotropic scans. So the resolution here is quite high. We can see more details than we normally would in a full brain scan. However, if I put it next to an OCT image that I'm going to explain later, which provides us with 3-micron resolution, you can appreciate how much further we can go and how much more information we can gain from this additional imaging modality when we are analyzing our MR scans. This table contains a set of regions of interest that are implicated in the networks that we want to study. And visually, just to show you where these are located. So this is an example that was manually segmented by Dr. Brian Adlow, and painstakingly analyzing and finding the location of all of these is a very time-consuming process. This is an example of a 22-week fetal sample. And we have to do this manual segmentation on various of our subjects in order to get a consistent representation of these areas and be able to study them. The next slide here shows you after manually segmenting the same areas and all of our currently obtained brain samples, we are able to seed the deterministic tractography or tractography solutions and see where they are connecting to what other ROIs or what other regions in the cortex they project to. So the examples here start from the fetal period. These are age ordered. So we have 3 fetal samples and then 4 perinatal samples and 3 adults, just to provide you with a baseline. The images show you deterministic tractography seated in the ventral tegmental area. And the screenshots are taken with a T2-weighted image in the background. The images that are framed are SID subjects. So what we are aiming to do and what we are currently studying is to compare the connectivity patterns in between SIDs and our, so to speak, healthy cohorts, and trying to anticipate differences that take place due to development and also what differentiates the controls from our SID subjects. Deterministic tractography is a very useful representation of our diffusion-weighted images. It helps us visualize the data sets very easily. However, this is the simplest model that we can fit to our diffusion-weighted images. So when we are analyzing our data, we use probabilistic tractography in order to keep a much richer set of information that's included in our images. When we run probabilistic tractography, we get information about how well different regions are connected to each other, and we can represent that information in connectivity matrices that I'm presenting you with here for the fetal and the postnatal and adult subjects. So each of these entry points in the matrices shows you a log connectivity probability, or how strongly some of these regions are connected. And on the left, and on the both axes, we have our 27 regions of interest that get manually segmented for this study. Now, looking at these matrices is a little bit difficult to make sense of. So we derive connectivity-based measurements in order to characterize. So these are scalar measurements that characterize the properties of these matrices. And we do comparison between different ROIs that are listed on the left, and also between the different aged subjects. Maybe a better or easier to interpret visual representation of this information is the following connectome animation. That's courtesy of Mark Olchani, who's a student of Dr. Brian Adlow here. So if you notice carefully and focus on the images, the ages of the subjects is increasing, and the width of these connections are getting stronger or getting lighter, depending on which phase we are at, as the age increases. So this is exactly the process that we are trying to quantitatively characterize and describe in case of healthy and SIDS patients as well. Now, in parallel, while we are doing this imaging and where we are trying to do this quantitative analysis, we also need to think about validating our data sets or validating our findings. And in the field, the ground truth is still described by histology. However, histology slices and images are destructive to the tissue. And these are hence not very accommodating when we are doing 3D imaging, where the slices need to be stacked back together in order to convey 3D information. So oftentimes, in order to guide histological 3D reconstruction, we take block face images, or these are actual photographs that are a picture or photo of the sample before the slice gets cut, so that any kind of tearing or folding is not going to be influencing the analysis of these images. The block face images contain definitely much more information and are less distorted than the histology images. However, segmenting them is still quite complex, and it doesn't provide us oftentimes with enough contrast to be able to analyze it. So as I previously alluded to it, we use another imaging modality, optical coherence tomography, in order to guide our validation and our segmentation of the MR images. Here, I'm going to show you a movie of the 3D stack of OCT images. The advantage of OCT imaging is that the imaging happens before the slice is being cut, just like it happened in case of the photographs. So there's no distortion tears, and the slices can be just stacked together because there's no distortion. We don't need to do any kind of spatial alignment or transformation between these slices in order to get the 3D representation of our tissue. The movie showed the 3 orthogonal views of the reconstructed OCT volume. Now this 20 micron isotropic OCT can be segmented in order to identify key anatomical regions. And then we are going to be able to focus on key regions, not only to guide our diffusion-weighted analysis, but also to help with the validation and relate characteristics of the tissue to histological findings in the literature. So this segmentation was done semi-manually. So every fourth slice of this OCT stack was manually segmented, and then we used an automated tool in order to fill in the rest of the segmentations in between. After our OCT imaging is done, we have the slices of the tissue. We can stain these with standard histological stains, digitize them, and now we have corresponding slices from histology and OCT in order to be able to stack them together into a 3D volume. The OCT volume is helping our restacking of the histology as well. And so we can come up, this is, for example, one of the reconstructed nickel-stained histology volume that we acquire after this alignment. And I'm not going to show you this movie because you're just a little bit short in time. And besides looking at, how nicely the histological slices align with our OCT slices, we can compare segmentations that are done independently of the two imaging modalities. And that allows us to validate OCT measurements, which, by the way, is the first time that we are using OCT imaging on such young subjects that are still myelinating. So we are discovering still a lot of features and a lot of interesting findings, but we can also state using these manual labels how accurate the alignment is between our different imaging modalities. And in order to tie all of this together, I'm going to show you a movie that brings all of these imaging modalities together. And that requires various spatial alignment tool development that I haven't really gone into detail here. But we started the movie with our whole brain MRI acquisition, which is 550 micron resolution. Then we zoom in into the brainstem MRI scan, which is 150 micron resolution. Then we are going through the OCT volume. And that's going to be soon interlaced by the histological slices. This is a little bit flickering due to the different staining of the histology sections, but you can, if you're focusing on the tissue, you can appreciate how well the histology and the OCT slices are aligned. Every now and then you can also see the torn histology tissue. That's really hard usually to deal with, but having the guidance of the OCT images, we managed to do quite well and robustly on this sample. Currently, the movie is showing you anatomical labels that replace both on the OCT and histological images that again help us in order to validate our findings, but also these ROIs are going to be seeds for our diffusion weighted imaging studies and tractography analysis. So this is a 3D representation of the OCT segmentation volumes, a 3D rendering of it. And the very last images are going to show you the serotonergic neurons that got localized in our images. And these are in red. They don't come out very well, but I'm happy to show you here on my computer. So these are important again in the studying of SIDS. The projects that I've presented to you are, a product of large sets of collaborations, both here at the MGH, but also at Boston Children's, University of Sao Paulo, Bonn, and Washington University. I listed my funding sources, and thank you very much for your attention, and I'm happy to take any questions.
That was great. The OCT is really amazing. What's the limit on the resolution? Is it 3.7 microns, or did you go farther?
We can go higher than that. However, when we want to acquire, three-dimensional volumes, it's basically memory and somebody babysitting the tissue in order to do the processing. So we can go higher. I think we had 1 micron resolution also. So in between 1 and 3, that has been the one that we acquired.
Did you combine that with any of those techniques to expand the brain? Used to increase resolution.
In order to incorporate OCT and with the
and you combine OCT with those like hair brain, expanded with polymers.
Yes. So the two different imaging modalities need different preparations from the tissue. So if you do, let's say, the clarity imaging, you can't really do OCT. But yes, there are several projects in progress right now where we are trying to increase the capability, or my colleagues are trying to increase the capability of imaging larger tissue samples. So here I showed you basically brainstem blocks that get imaged. So the idea would be to have a full brain slice image. And as I showed you, we are already able to register these in together with other modalities. So the idea is to fuse all sorts of information from different imaging modalities in order to guide our analysis. Any other questions? I'm gonna look online. I think I got the, so there's one question here about, so there's a question about these techniques look extremely exciting. What will you use as control group in your study looking for causal lesion in SIDS? That's a very good question. So many of our, so to speak, control subjects come to us after drowning. So, and also we are collaborating with several hospitals that can identify infants who pass away with non-neurological reasons. But there's a very heavy kind of screening process that kind of establishes our control and the SIDS cohort. Okay, I think this is it for questions. So unless there's any more from the audience, thank you very much for your attention.